In this video, an acid-base titration will be performed. Before we begin, it is important to understand the purpose of a titration. Titrations are used to determine the concentration of an unknown substance in a solution using a standard solution. In this experiment, we are trying to determine the acid concentration in a soda using sodium hydroxide as the standard solution and the soda as the unknown solution, which is called the analyte. Before beginning the titration, the sodium hydroxide solution was standardized with weighed samples of KHP in water. This information will be needed in order to calculate the concentration of the unknown at the end of the experiment. Then the soda was heated for 15 minutes to eliminate carbonation and then added to an Erlenmeyer flask with 2 drops of the indicator phenolphthalein. This indicator will allow us to see the endpoint of the titration as a light pink color. It is important to prime or clean the burette before the titration 3 times with water and 3 times with sodium hydroxide. We can now begin the experiment. Once the 3 drops of phenolphthalein have been added to the solution, we can read the initial volume of sodium hydroxide in the burette. When reading the volume, it is important to be at eye level and to round to 2 decimal places. Once this initial volume has been read, we can begin the titration by slowly adding sodium hydroxide into the solution, being sure to swirl the flask as we do this. Now that we see the endpoint of a faint pink color, we can read the final volume of the burette. Once again, this is done at eye level and recorded to 2 decimal places. Trial 2 was done in the same manner, keeping in mind that all volumes are taken to 2 decimal places, reading the meniscus at eye level. Once again, the drops of phenolphthalein are added in order to ensure that we will see the faint pink endpoint at the end of the titration. Now that the solution is ready, we can begin the titration by slowly letting out the sodium hydroxide into the flask, being sure to swirl the solution. As you can see, just a few drops will transition the color to its endpoint, so it is important to go very slow at the end of the titration in order to ensure just a faint pink color. In this picture, you can see the difference between a faint pink color and a clear solution. Now that we have achieved this faint pink endpoint, we can take the final volume of the burette and once again being sure to be at eye level and recording it to 2 decimal places. Finally, the third trial was done, once again adding the drops of phenolphthalein and swirling the flask in order to ensure that the indicator was well into the solution. Now, the initial volume was taken at eye level and at 2 decimal places. Once this volume was recorded, sodium hydroxide was released from the burette into the flask, being sure to swirl the flask. As you will see, just a few drops will make the difference between a color change. As you can see, in comparison to a normal solution, there is a faint pink color. Therefore, the final volume of the burette can be taken. This middle flask is a preferred faint pink color. And now that we know our KHP mass and our initial and final volume, we can determine the unknown variable.